Part 4 of Chapter 5 is important because there are a lot of things we need in order to get the nutrients. It's all great to know that we need nutrients and what kinds and how many calories and so on, but ultimately we need to figure out how we can get them into and out of the cell. And so this fourth part includes two types of transport. The first of these is called passive transport. And the reason why it's called passive transport is because here we are not actually needing any additional energy. This is just based on the laws of physics again. Now we know already that molecules contain potential energy in their bonds. So this is something I already mentioned. But it turns out that these bonds vibrate. And because they vibrate, this is something that makes the molecules move even without any additional excitement. So molecules, they're a little jittery, kind of. And so this vibration ultimately is something that makes them move randomly and bounce into each other. Water molecules are like that. All kinds of molecules have this capacity, and it's really very clear to see when you do a very simple experiment with maybe a warm glass of water and a sugar cube. So you take the sugar cube, you put it in the bottom of the glass of water, and eventually the sugar will all by itself just sort of fall apart, and it ends up stirring itself, literally, because of the random movement of molecules. And that is what's called diffusion. So if space is available in, let's say, the liquid or the gas, the molecules may move to fill it. And because there is no energy needed to do this, because it's an intrinsic thing that these molecules do, we call that passive transport. So passive transport without energy. Now here are two examples for what happens with diffusion. And in each case, you can see that there is a certain concentration on the left, a concentration of certain substances here and here, and there is some kind of a membrane in between. So this thing, this yellow here in the middle, that is actually a membrane. Now, when you have this situation, you'll find that these molecules of dye, they're going to bounce around, they're going to bounce into each other, they're going to push each other, just if you had a bunch of marbles that you're rolling around, and eventually one of these is going to pop across that membrane. And so here there is a certain amount of diffusion that goes on from the left to the right. That's where the direction of diffusion is. And once you get to a point where there are as many molecules here as there are over there, then you've reached what's called equilibrium. So the concentration on one side is the same as the concentration on the other, and then they will still bounce, but there's going to be no particular direction for which way the bouncing is going to go. I think most people have no problem in understanding this one because that's intuitive. But what's really interesting is when you use membranes and when you use different molecules, it also works when you have different kinds. So the red ones are at a high concentration here and at a low concentration there. So the red ones are going to move to the left. The blue ones are going to move to the right until both of them have reached equilibrium. And this is what diffusion does. Diffusion will keep going until on both sides of this membrane, or in both parts, in all parts of the solution, there is a mixture that is in equilibrium. And so this is a very interesting concept, because what it means is that you don't have to mechanically move things from one point to another. Things will just flow. Now there is a special case that you can construct, and this is important for our cells, whereby you look at what happens when you create a membrane that's kind of like our cell membrane and put it in the middle of the solution. So here you have the solution. This happens to be water, and there is sugar at two different concentrations on either side of this in the U-shaped test tube. Now, U-shaped test tube means that the water level on both sides is going to be the same. So the water level over here is going to be the same as the water level over there. The sugar molecules would now like to move so that there's equilibrium, right? So some of these green sugar molecules would like to move over to the left, in order to achieve equilibrium. But because we have this thing called a selectively permeable membrane, which is basically what our cell membrane is, they can't. So what are they going to do? Well, they can't do anything. But the water can move. The water molecules are small enough that they can move. So rather than having the solute do the equilibrium, it's going to be the solvent that does it. And here's the really neat thing. If you do this kind of an experiment, you would expect in a U-shaped tube to always have water at equal levels, like here. But once you put the membrane in there and you create the situation, the water will actually rise in one arm of this U against the force of gravity. That's really critical. The movement of these molecules is so strong, the desire, if you will, to create equilibrium is so strong that it can overcome the force of gravity. Now that's no mean feat. I mean, think about this. If you wanted to try to overcome the force of gravity, you'd have to jump. That requires a lot of energy. And if you want to do it more, then you'd have to fly. So that's not so easy, right? And so here, these molecules, they can do it. They go quite a ways up. This water can drive this concentration, and ultimately you reach equilibrium. And that's osmosis. Osmosis is a special kind of diffusion. It's a diffusion where water moves. And this is one of the things that, of course, can happen in our cells. We can achieve equilibrium with things inside and outside of the cell because we have water inside and out of our cells. So some of those things, that's very useful. As a consequence of the movement of water, you can sometimes see concentration gradients in the cell, where on the inside of the cell you have a different concentration than on the outside of the cell. And so if the cell has a low concentration of something, low salt, low sugar, low solute, whatever it is, then on the outside the concentration is hypertonic. So the cell is actually going to be in a hypertonic environment. Now, I'll show you a picture of this in a minute. I just want to get the vocabulary down. And if the opposite is true, if the cell has the higher concentration and outside the concentration is low, then the cell is in a hypotonic solution. Now, most people know these prefixes, hyper and hypo, because it's a blood pressure thing, right? So if you have high blood pressure, what is that called? Hypertension, right? So hypertension is high blood pressure. Hyper is high. Hypotension is low blood pressure. And when the outside and the inside are at the same concentrations, then this is an isotonic situation. Now, this the example I showed you a minute ago, osmosis, is what creates an isotonic environment on either side, if you will. It creates equilibrium on either side of this membrane. And osmoregulation is how our body affects that. So if you end up being dehydrated, that means your water balance is too low. You need to drink some, and then your osmoregulation will take care of balancing the water out again. Now, when you look at this in a picture, you'll see that the external environment can really do a lot to a cell. So in the first case, you can see there that the cell is in a normal environment. And this is here indicated both by the shade in the background, the shade of blue, and what the cell actually looks like. So this is a red blood cell, and the red blood cell is in a normal environment, and so the amount of water that goes in is the same as the amount of water that goes out. That's normal. Now, if you place the cell into a hypotonic situation, like pure water, then you find that more water is going in than out, and as a consequence, the cell could literally blow up. The opposite is also true, that if you have a high concentration on the inside there, that is a hypertonic situation. then you'll find that the water is leaving the cell to try and create equilibrium on the other side. Well, it's not going to, because if like an ocean has a high concentration, there's not an amount of water from your cells that's going to reach equilibrium. So the cells are simply going to be left high and dry. They shrivel up, and that's, of course, not very healthy. So if you eat something that's very salty, it doesn't take very long before you feel the need to have a drink. And that's because some of your cells are starting to experience this situation. And so your body can react to that by osmoregulating. Now, in plant cells, there's a similar situation. The concepts ultimately are still the same. You have the isotonic solution, you have the hypotonic solution, and you have the hypertonic solution. But because plants have things like stems and leaves that don't have skin like we do or bones that we do, those plant cells are going to be the support of the entire plant. And so they have a cell wall. That's what this is, the cell wall. And because they have a cell wall, they need to make sure that everything gets propped up. So plants actually need to be in a situation where they can access pure water. That's why you put plants into water when you display them. They want a hypertonic solution so that they can have enough turgor pressure to stay upright. If you have something like this, you put the plants at equilibrium, you find that the heads of the flower, they may droop very quickly, and that's simply because the pressure isn't enough to keep it upright. Whereas if you put them into an environment like this, then they can remain upright. But think about this for a minute. If you have cut flowers and you Valentine's Day, you give your flowers to somebody and it's great, and the next day the heads droop, what are you going to do? Well, one thing you can do is fill the sink with water and immerse the flowers in it entirely and leave them overnight. And you will find that in the morning, with all the different places where the water can enter the plant cells, you're going to have that turgor pressure back, and so you can save those flowers for a little bit. So let me tell you, I'll save you money here by telling you this. Now, active transport is something where energy is required. Energy is required, and so when you look at this image, you can see right away, there's ATP. And so here is the case that we saw already when we talked about ATP. This is your phospholipid bilayer, so you have the phospholipid heads and the phospholipid tails, and there out here is something that the cell would like to have, but the molecule is a little bit too big for it to just float through the cell membrane, and so what you need to do is bring it in. How you bring it in? You use a little phosphate group. The phosphate group is going to be able to be stuck on there, and then when the phosphate group pops off, it gives energy to this transport protein, and you end up being able to bring in the solute. Now, it's not only about size, by the way. Solutes are sometimes needed to accumulate in a cell. How are you going to do that when your body is always trying to reach equilibrium? Well, you have to go against equilibrium. And so in this case, you can see that the concentration down here, this is the symbol for concentration. The concentration down here is already high. The concentration here is low. And so normally what you'd expect is that these solutes would like to go out there. But you don't want that, because you want the sugar in your cell. You want the ATP in your cell. So you don't want this to happen. And the only way you can prevent this from happening is to put in a membrane that keeps things inside, and to have ways to transport things into the cell in addition, against this concentration gradient. And that is what active transport is needed for. So it's bringing things in that are bigger, and it's bringing things in against the gradient. And that's how you accumulate food. There are two main ways to move things into and out of the cell. One of these is exocytosis. That's how you move things out of the cell. And exocytosis includes, as you can see here, some kind of little vesicle. These vesicles are the things that we described earlier in chapter 4. And one of the things we said is these are the kind of things that transport protein precursors from the ER to the Golgi apparatus. And then they transport things from the Golgi apparatus to the outside of the cell. And as you can see here, they have a phospholipid bilayer, and they simply merge into the plasma membrane. They merge just like we saw with the ping pong balls in the baby pool. That's how they merge in, and then once they're merged in, they can release the waste products that need to be released. The opposite also works. If there is something really interesting that's coming close to the cell might be stimulated to fold inward like this, and when it folds inward, it brings in some of the substance. It's like, sucking something in with your mouth. You suck it in, it gets in there, and then all you have to do is close it off on the top, and if you do it, then you end up being able to bring in these substances into the cell. Now, this is when you're bringing in a whole bunch of stuff. When we looked at active transport in the previous slide, it was one molecule at the time, and that of course also works. But sometimes, especially when it comes to food, if there is a lot of food available, might as well use it. And this is the kind of thing that can happen. Now, when it comes to bringing things inside of the cell, there are different ways by which different substances can be brought in. One of these is called phagocytosis, and that is cellular eating. Anything cytosis, it has to do with cells, and this is another lesson in Greek. Phago has to do with consuming, eating, and that's what we do here. So here we have cellular eating. And in this case, you can see that there is the food. This is a single-celled organism called an amoeba, and this amoeba literally slimes its way around the food, and it will then be able to bring it in. So this is cellular eating. It moves across the substances. Now this is three-dimensional, this picture, so it happens from above and below. It's kind of like the blob, it slimes over everything. And that's what amoebas do. Our cells are very similar. Our cells, when you look at them individually, they also move kind of like this. The second of these is called pinocytosis, and pinocytosis is cellular drinking. Cellular drinking, when you have droplets of fluids or oils that are being brought in, they can be brought in not by engulfing, but by literally merging with pieces of the membrane. When you think about cellular drinking here, this is not drinking water, because water pretty much can get in and out of the cell relatively easily, but this is other kinds of fluids, and lipids are one of those. And a lipid, well, the phospholipid bilayer has an affinity for lipids, and so lipids are being brought in a different way than solid foods. And so this is cellular drinking. The third type is the most specific type of active transport, and that's called receptor-mediated endocytosis. Receptor-mediated, what does that mean? Well, a receptor is something that you have on the surface of a cell. And so on the cell surface, you're going to have like a little flag, and on this little flag, you're basically able to catch certain kinds of molecules with it. Endocytosis is to bring things into the cell. So it brings things this way, brings things into the cell, but using a receptor, and this receptor only brings in particular types of molecules. It brings in particular types of molecules because those may be the things that particular cell needs. There could be a variety of those kinds of cells, and the cell can actually use some of these in passing signals from one cell to the next. Only when the correct molecules is encountered, then the molecule is bound and interacted with. So it can be a particular food molecule, perhaps. It can be a signaling molecule. And if you think about something like perhaps a nerve cell, that's a good example for cell-to-cell signaling, you'll find that at the end of one of the nerve cells, you're going to have chemicals, and these chemicals are going to be released when an impulse travels. So here is the impulse. It travels and it hits the end of this nerve cell. And this nerve cell now releases a variety of chemicals. It releases particular signaling molecules. On the other side of this little gap is another cell. This is the cell that's going to receive the stimulus. And here it has these little receptors. And so the molecule from the one cell hits the receptor, and that is what then allows the signal to travel on. Now think about this for a minute. This is something that happens with remarkable accuracy and remarkable speed. Because if you put your hand on a hot plate, you're not going to sit there and go, right? It's more like instantly you're going to pull this thing away. And so what has to happen from your finger or from your hand, the stimulus has to travel through the nerves all the way up to your brain, and then the brain is going to go, this is hot. And it's going to send a signal all the way back down into your muscles, and the muscles are then going to pull the hand away. Now, the way I just talked about it, that takes way too long. By that time, your hand would be really burned, right? And so that's obviously not something that happens. It is almost instantaneous the way this signaling works. And that's how receptor-mediated endocytosis can help us. And we have a variety of these signal transduction pathways. They are sometimes fairly complex, and they allow us to steer certain kinds of reactions in the body. And if you do these kind of, if you study signal transduction pathways, you realize how efficient they can be. And in fact, when you're an athlete, you can sometimes train to be better on those. You can fine-tune your body so that you're able to deal with these kind of signals well. And that's one part of becoming an athlete, is to start to control somehow how these signal transduction pathways prepare you to be more efficient in how you run a race or how quickly you get out of the starting blocks and so on. All right, and that's all we're going to do for this slideshow.